Good morning, and thank you, Jay. And thank you, Bruce, for this wonderful symposium. So these are exciting times for targeting the PI3 kinase pathway in cancer. This is a pathway that, as Bruce has mentioned, has been extensively discussed over the last few days. And the reason why it's perhaps so prominent is because it plays a role in a large majority of tumors that we deal with, and in particularly has a clear role in breast cancer. This pathway is aberrantly activated in a large proportion of breast tumors. The mechanism by which this pathway becomes aberrantly activated, there are multiple mechanisms, so it can be HER2 overexpression for one. It can also be mutations at several sites of the PI3 kinase gene itself, PTEN deletion, AKT mutations, etc. So, and I think it's probably true that whenever a pathway is frequently mutant in cancer, is that it plays a driving role, and that's the case. The roles of the PI3 kinase pathway are multiple. They control the input from many of the receptors. They control cell metabolism and many other functions. So again, a critical pathway in many respects. The way I became interested in the PI3 kinase pathway was because a few years ago, when we were working exclusively on anti-HER2 therapies, we began to realize that there were a proportion of tumors that overexpress HER2 that were not responsive to anti-HER2 therapies. And this happened in the clinic, and there were a number of papers published in that regard. And in the lab, this was something that was easier to demonstrate. So initial work by the group of René Bernards in the Netherlands Cancer Institute showed that very clearly. And actually in our group, a postdoc that had been with René in the past, Piet Einhorn, began to work on this as well. And the data were extremely consistent in the number of labs that were addressing this issue. So namely, those cell lines that were engineered to overexpress the activating mutations of PI3 kinase were resistant to anti-HER2 therapies. And you have here data both with trastuzumab, in which you can see the cell lines with these activating mutations are resistant, and also with the small tyrosine kinase inhibitors such as lapatinib. So this was our entry point, if you wish, into this field. But then we immediately realized the tremendous opportunity that it provided as a potential place to develop therapies against. And today we have a number of approaches to target the PI3 kinase pathway, ranging from PI3 kinase inhibitors itself to AKT inhibitors to mTOR inhibitors, and then to a combination of them. What I'll do is I'll go by chronological order. When we began to be involved in this, the only compounds that were available for clinical trials were the rapalogs, and we began to work with everolimus. And I will not show you the phase 1 data, although I'll refer to some of the findings that we found later on. But I think what we observed, and that was quite interesting, is that everolimus, which is a rapalog that, as you know, blocks mTOR1 function, so it's not a good blocker of mTOR2. Everolimus had some activity in breast cancer, but not much. This is the base data that you will find in breast cancer with everolimus. This is data from the NCIC Canada. So if you guys are from any drug company and want to have a positive trial, go to Canada because this data has not been reproduced by any of us. But again, even in a very good setting, the response rates that were observed were very minimal. So this is data from Ellard et al., and showing that they had a very small number of partial responses. Interestingly, some of the clinical investigators dealing with everolimus had the feeling that some patients actually got worse when we treated them with everolimus. And I don't know if Neil, I wasn't here when Neil gave his talk, but Neil has experience in prostate cancer in which some patients did get worse on everolimus, as if you were activating other pathways that render the cells not only resistant, but actually activated upon blockade. So this is data on the activity. And then another important component of this trial is the fact that the responses that they observed, and this is a busy table, but just focus to the CR and PR, complete remission and partial response, and the responses were seen basically in those patients that were ER positive. So we're beginning to see a few things that are interesting. Everolimus has little activity, and activity is seen in this subset in patients that were ER positive. So I think what's happening here, and we have some clinical data to support it, is that there is a profound crosstalk between the estrogen receptor and mTOR signaling. And there are several lines of evidence in support of that. The first thing that we know is that mTOR activates ER signaling in a ligand-independent fashion. So all these ligand-independent mechanisms of ER activation, many of them go through mTOR. So that's point number one. Now, point number two, in the lab, if you treat cells that are ER positive with an mTOR inhibitor, okay, you may see apoptosis. Now, that apoptosis is rescued, and that's work from Mike Ellis at WashU. That apoptosis is rescued if you provide estradiol. So ligand-dependent receptor activation might rescue apoptosis induced upon mTOR blockade. So that speaks for that interaction. The third point here that I'd like to mention is that in clinical samples and in cell lines, and this is data from Carlos Artiaga's group and others, hyperactivation of this pathway is observed in endocrine-resistant cells and also in some tumors. So I think if you put all this together, mTOR plus an AI blockade perhaps is the way to go forward. And that's exactly what we proposed and what we did. So this would go like this. You block with an AI alone, and that may be insufficient in situations where you may have strong PI3 kinase mTOR signaling that leads into into ligand-independent ER transcription. So that's situation number one. Number two, if you block mTOR signaling and you leave estradiol-dependent receptor signaling undisturbed, that it's overcome and you still have ER transcription. So perhaps the way to go forward, and that we propose, is that you have to block the two pathways at the same time. And in order to prove this, we conducted a randomized neoadjuvant study in breast cancer. Why neoadjuvant? In breast cancer, we need to be able, and breast cancer and many other tumors, we need to have biopsies to see what's going on. And it is very hard to do these studies in patients with advanced disease. They are not homogeneous. It is difficult to get. You can get some biopsies, but you cannot get 200 biopsies. So we have been implementing for the last few years neoadjuvant approaches to new drug development, and it has been extremely fruitful. And in this case, what we did is that we had patients that had ER-positive early breast cancer were randomized to receive AI, letrozole, an androgen inhibitor by itself, plus placebo, versus the same dose and schedule of the AI plus everolimus, also known as RAD-001. And the primary endpoint of the study was to look at response rate, but even more important was to look at tumor biopsies on day 15 and compared with the pre-therapy biopsies and see what was happening at the level of proliferation. The study was positive, so we observed more responses on the everolimus arm. But to me, the most interesting thing is looking at the decrease of proliferation measured by Ki-67. In this graph, what we have here is those tumors that have very low proliferation when we look at them. So in basal samples, you have this distribution, which is the classical distribution of proliferating tumors. So you have some tumors that have very high proliferation and some that have a very low. Day 15 on AI alone, you have about 30% that have undetectable levels of Ki-67, and you double that when you add everolimus. So this was a very strong indication that this was going to be synergistic in the clinic in support of the in support of the preclinical work. We launched a phase 3 study that, as you know, phase 3 studies in breast cancer take a long time to be enrolled and to be designed. And when we were fully enrolled with our phase 3 study, and you'll see that in a minute, we were extremely anxious about whether the trial was going to be positive or not. And we got a very positive psychological boost, although the trial was already finalized, our trial, when last year there was a small phase 2 randomized trial reported. That's the TAMRAT study. So these were in patients that had postmenopausal disease, ER positive, and they were treated with tamoxifen plus minus everolimus. All these patients had received a prior AI. And this study was very positive. Again, small study, but very positive. Look at the hazard ratio, 0.53. And so we felt good. We said, maybe our study is going to be positive as well. And that's the design of our study, and that's the BOLERO 2. That's a registration study in patients with metastatic disease, 700 patients, randomized 2-to-1 to an AI. In this case was an steroidal AI, exemestane, plus placebo versus everolimus exemestane. Why do we do 2-to-1? This is something that we increasingly do in our clinical trials. We want to make sure that a higher number of patients get exposed to the experimental arm, especially if you have a suspicion that it might be better, and it's absolutely fine to do it. So this facilitates enrollment and makes also the protocol more interesting. And the primary endpoint of the study was progression-free survival. Also, we had overall survival, bone markers, which is very interesting, but I will not have time to talk to you about this, safety and PK. Patients were stratified based on the prior sensitivity to hormone therapy or visceral disease, and importantly, there was no crossover allowed because we want to see what's happening with survival. And the primary endpoint of the study, was to have a 26% reduction. That's a hazard ratio of 0.74. So we would have been happy, actually very happy, if the study would have shown us a hazard ratio of 0.74, which is a very reasonable endpoint in a randomized phase 3 study. In order to do that, we needed this number of events, and importantly, the study had one interim analysis when 60% of the events occur. And these interim analyses, they are very stringent. In order for a study to be called positive by the pre-specified interim analysis, they have to cross a very stringent boundary, as described by Fleming, and that's the p-value. So it's not easy, it's not stringent. But anyway, the good news is that the Independent Data Monitoring Committee let us know on February 11 that these pre-specified boundaries have been met. So the study is final. I mean, the data is absolutely final, and that's what we got. So the primary endpoint of the study by local assessments, by local investigators, was progression-free survival, and we went from 2.8 to 6.9 months, and that's a hazard ratio of 0.3. You have here more zeros that you can care for. There are 15 zeros on the p-value. I know that statistically, after more than 3 zeros, it doesn't matter, but boy, it makes you feel good to have all these zeros there, just because of lack of knowledge. And that's based on the local review. And then the central review, which usually is worse than the local review, in this case is even better, and the hazard ratio here is 0.36. 10.6 months. There is not a single clinical trial in metastatic ER-positive breast cancer that has this data. And this is possibly the most positive breast trial in this setting. And I think that's quite exciting for the field because, again, this is an imperfect compound. I mean, this is nothing. I mean, this is like version 1.0 of what's going to be a much better number of compounds. Who benefits from this? We did a sensitivity analysis and we look at different subgroups, and basically we saw benefit across the board. So it doesn't matter whether patients had visceral disease, it doesn't matter whether the patients had prior sensitivity to hormones. So I think probably here, looking at this magnitude of the effect and looking at the fact that everybody benefits, we have here perhaps a combination of luminal A's, luminal B's, I would think. So it's quite uniform across. And in this self-serving slide that I've taken from Fabrice André, who discussed this at the ESMO meeting, The point is that people will ask, how good is this data? Well, here Fabrice put up some of the classical targeted therapy trials and look at the hazard ratio. And our hazard ratio is right there. So our Trastuzumab trial that showed the hazard ratio of 0.51 and was what got Herceptin approved ours is even better, 0.36. So I think this is, if you have to put this in perspective, is something that has a lot of value. Now, so we have seen that there is a crosstalk between ER and mTOR, but there's also a lot of crosstalk between PI3 kinase, mTOR, and other pathways. This is work done with Neil Rosen, and Neil is the one who began to look in cancer cells and looked that upon rapamycin therapy, there was an activation of upstream AKT. And the basis for that is that upon blockade of mTOR, there is a negative feedback loop that is S6K dependent, that is released, and then IRS1 levels go up, and that activates signaling via the IGF-1 receptor, which in turn to increase AKT. That is the basis, I guess, for those patients that got worse. You could imagine that you're blocking mTOR, and in some patients you have an AKT activation that drives proliferation per se. So I think that has value. In clinical samples, we were able to see the same. This is data from our phase 1 study done by Josep Tabernero at Vall d'Hebron, and you can see the AKT inhibition in 2 samples here, in 2 biopsies, pre-therapy and on therapy, so quite clear data. If IRS1 is the mechanism of activation of the feedback loop, what happens if we block IRS1 signaling with an antibody against IGF-1 receptor? And that's exactly what we did, and we didn't do it in the lab. Yes, we did, but we also did it in patients. And this is data from patients we just published in CCR showing that we were blocking phospho-IRS1, phospho-AKT, and phospho-MAP kinase in patients treated with this antibody. So clearly, this was a good thing. And then the other thing that is important is that there was some data, and this is data from Merck, showing that in tumors that are, these are multiple cell lines, here you have ER expression, and here you have the Rasnes bias gene signature of the cells, and it shows also that Ridaforolimus is another mTOR inhibitor is also more active in cell lines that are luminal B. So again, suggesting that this mTOR dependency may be more pronounced in ER positive cell lines as opposed to triple negative. And this is data from the Karolinska showing that this follicle available, showing that looking at levels of IGF-1 receptor expression that is higher in tumors that are ER positive with a high growth proliferation index. So these are the luminal Bs. And then data from Merck that has not been published, looking at enhancer screens, no matter what the screen they do with Dalo or with Rida. So with Dalo, which is the IGFR antibody, every single time mTOR, FRAB1 mTOR is a top hit, and the same happens in the reverse. So again, everything was getting together in supporting that there was also a crosstalk between IGF-1 receptor and mTOR, and we did a phase 1 clinical trial with the combination. There is a phase 1 with multiple doses, escalating doses, so not everybody got the right dose. And I think what's important in this phase 1 study is that we observed that 54% of the patients with ER positive hyperproliferation had some evidence of clinical response. And in many cases, this was a partial response. So now we are doing a large randomized phase 2 study of this combination. We are seeing responses, and I think this could be yet another strategy. And this could be the only way, at least in the near future, to get IGF-1 receptor antibodies into the clinic in breast cancer in combination with mTOR inhibitors. We are now moving ahead. We have a list of compounds. This is a partial list. This is just a list of the compounds for which we are doing clinical trials that we are personally involved. So there are many more, of course. And as you can see, there are different target specificities. So we have alpha inhibitors, we have beta-sparing compounds, pan-PI3 kinase, PI3 kinase mTOR, mTOR catalytic, and AKT inhibitors. So we have a whole variety. Which ones are better? We have no idea. We have done some work that I'll show you with one of the pan-PI3K inhibitors, that's BKM120. That work is in press in JCO, and here in this study, what we did is that we had multiple tumor biopsies, and we were looking at inhibition of downstream from PI3 kinase, and looking at phospho-S6 and looking at phospho-AKT in a number of patients. We did see inhibition, so we are blocking the pathway, there is no question. In the clinical trial, in the dose escalation phase, with a PET scan companion component, and something that is important is that in a majority of patients, we did see a PET scan decrease, a PET scan response, if you wish, a decrease in glucose uptake. Now, the important thing here is that in a proportion, in those patients that had a radiological response, and we'll see them in the next slide, all of them had a PET response up front. So one thing that one could envision is that we perhaps could use PET scans as a way to identify early on who is going to respond to this or not. And if you don't have a response by PET scan, say on day 15, the chances that you will respond to one of the lesions is very limited. The other one is looking at in the dose escalation phase at the activity. So we had some activity, nothing to write home about, but we had some activity seen with this compound. Interestingly enough, the best response we had was in a patient that had breast cancer and had a KRAS mutation. And this is very different to the very poor performance of patients with colorectal cancer that had KRAS mutations and they do not do well. So maybe in breast cancer, a KRAS mutation that is very rare has a completely different functional meaning than in colon cancer. Once we find the dose, and we had now an expansion cohort, and we are seeing activity in HER2-positive disease. This is a patient of mine with a major response by PET scan. And also we began to see responses in ER-positive disease. And here you have a PET scan prior to therapy and on therapy, and then the huge response seen by CT scan. You can even see that the liver is actually a little smaller in size because of the tumors really decreased dramatically. The compounds that we're really excited about are these alpha-specific inhibitors. And we're working with the three of them. These are two alpha-specifics, and this is a beta inhibitor. And we are excited for one reason. These are the first compounds that we see selectivity based on PI3 kinase mutational status. So this is work from Cyril Burns at the CMT at MGH, and Novartis has the same data working together with the Cell Line Encyclopedia. And in those cell lines that have mutation of the PI3 kinase alpha, the sensitivity is much greater. Now, it's interesting the way clinicians and the way lab guys look at the same graphic. When we saw this data in the lab, the clinicians in the group said, this is fantastic. We're going to be able to enroll patients with PI3 kinase mutations. The guys in the lab said, who are these guys? Who are those that have sensitivity and are not having a PI3 kinase mutation? So what we said is, you guys figure this out. And in the meantime, if you don't mind, we'll proceed with those. And that's what we're doing. So that's what we're doing. Okay, so we are enrolling into this trial. From day one, we have entered patients that have PI3 kinase mutation. This trial has been run by a fellow at MGH, in Zurich. He has enrolled more patients himself than anybody else in 6 other sites around the world. And that speaks to the strength of the MGH fellows, right? And we have seen responses in patients. So this is working clearly. And we also seen responses with the other compounds. Now, one last concept, and that's something that Neil touched upon. I'm sure he did. And that's, so we have seen that you activate pathway compensatory mechanisms when you block mTOR1. What happens when you block PI3 kinase? Well, the same occurs. So in multiple cell lines, we have seen that when you block PI3 kinase, you have an increase in phospho-ERK. These are two cell lines that are HER2 positive, and this is not compound dependent. So we have taken P110 inhibitors, P110 mTOR, AKT, mTOR catalytic, mTOR1, mTOR catalytic, and in every single case, you have an increase in phospho-ERK. And in the case of HER2 positive cells, this goes along with phospho-HER2 activation. And Neil and ourselves, working together, we have shown that this is through a FOXO-mediated upregulation of HER3. So there is a transcriptional increase in HER3 in addition to activation of HER3, and that basically in HER2 positive is what drives this feedback loop. So you can envision several situations where you have an IGF-1R driven loop, a HER2 driven, And EGFR receptor-driven, that's work that has been done by the group of Jeff Engelman, showing the same. So there's a consistency here. And I think what's exciting is that this gives us the opportunity to use combination therapies to block this activation. So you can see you could block HER2, HER3, or you could block ERK as well. We don't know what's better. In the lab, it looks very similar. And in the clinic, we are doing the clinical trials together. We're also working together with Jeff Engelman's group and Bill Hahn's group and the group in Barcelona in trying to identify mechanisms of resistance to PI3 kinase inhibitors. So we're trying to be ahead of the game because we are convinced that these therapies will reach the clinic. So we have several strategies that I think for time purposes I'm going to skip. But basically, we are beginning to have some hits that make sense and that we are going to be able to identify in our patients as we move along. So I think I'd like to leave you to today is that this pathway clearly is important. We're beginning to see responses in patients. The rapamycin analogs will clearly be part of therapy in ER-positive breast cancer. People are talking about launching adjuvant trials. People are talking about doing first-line settings. So that's going to change and going to be a new standard, I'm sure, in ER-positive disease. And that we have to then move in with the new, more specific compounds that are very exciting. Combinations is going to be the name of the game, combinations that are rational. So I am personally very excited about the anti-IGF-1 receptor and anti-ERK. And then also the combinations blocking PI3 kinase and HER2, and perhaps PI3 kinase and ERK as well. So I think we're going to be busy in the next few years. So I'd like to thank the people who have been working on this. In the picture, if you can see them, you have Mauri Scaltriti, who's leading my lab, and he's here today. We have our super fellow, Diane. We have Pete Einhorn. This is the group in Barcelona that has done a lot of the work. This is Josep Tabernero, and this is Jordi Rodon and Violeta. And the guys here that I'd like to acknowledge is Neil Rosen and Sarat from Sloan Kettering, and then also Jeff Engelman, Cyril Benz, and John E.F. Freti for all the work. And this is my funding. Thank you very much.
Great talk, as always. Just a question on the BOLERO trial. So first of all, fantastic results, obviously very exciting. When you see a clinical trial result, particularly a phase 3 with a p-value with 15 zeros, obviously everyone runs for the champagne. But it also makes you wonder, in the back of your mind, how overpowered was the study? And are we now, should we start thinking about clinical trial designs that are much more adaptive so that that trial could have been done with 100 women and not 700 women as the data is maturing, because it would have been statistically significant with far less number of patients, but obviously you didn't know that when you designed it. So are we moving into that era in terms of adaptive designs that would decrease the number of patients needed for these trials?
Yeah, I mean, you're so right. So I think even the most optimistic person would not have predicted these results. I remember we had an advisory committee of the people working in this, and this was a few months ago, and the whole theme of that meeting, talking about BOLERO-2, was how to use the BOLERO-2 samples to learn on how to use PI3 kinase inhibitors, because the investigators had the feeling that BOLERO-2 was going to be marginally positive. And at one point I had to tell them, guys, could you please wait until we see the data? So this was unexpected, even for all of us. So that's one thing. Now, if you do adaptive design in the metastatic setting, I guess it's a balance of time and effort. So, I mean, how would you do that? Unless you get approval I think the only way you could get by that is that if you could get approval to do studies like in the neoadjuvant setting, if we could have a way to get approval there, and then I think we'd be okay. But in the metastatic setting, I see it very hard because I see it hard. I don't know how I would do it because of the time-sensitive nature of what we do.
Jose, what about those samples you said the laboratory guys were going to pursue? Did they ever find out what was different about those patients who were so sensitive?
Right. So IGF-1 receptor plays a role there, clearly. So cell lines that express IGF receptor, yes. So that's one. And that's what we know for sure. And that's, yeah.
So could you select those patients to be part of the trial as well?
We could. But I think our obligation right now is that since the alpha-specific compounds are so clearly working on alpha-specific, I think if we could get that settled. I think the idea is to get that concept proven, and then we're going to have the chance to work on the non-PI3 kinase mutant.
Other questions? Just one other question. You mentioned the possibility, I mean, the results are so striking, the possibility of using an mTOR inhibitor or a pathway, PI3 kinase inhibitor, with anti-estrogen therapy as primary treatment. Where does that stand? I mean, it seems so logical now that if you find it in resistant disease, it should be true.
Absolutely. So I think what needs to be done, and quite urgently, is to study this in the first-line setting, and then most importantly, in the adjuvant setting. I mean, that would be, I mean, this would be like the end of chemotherapy for ER-positive disease. I mean, this level of magnitude. And also, it's important to realize that these tumors are not very responsive to chemotherapy. So I think that what's happening in the field is that we all very actively trying to come up with a very strong adjuvant design. And I think that's where this could play a role. And then, even in prevention, right? But at this point, yeah.
Right. Has that started yet?
Well, the data were presented on the 26th of September. Since then, I know that we have 6 proposals on the table. It's not bad. We have like a proposal per week. So we'll see.
No one plans for success, huh?